Nope. Let's go that way first. All right, so I want you to go ahead and fill in your blanks for that first slide. Okay, so one last thing with your cholera case study. Make sure you're reading all the text. I obviously want you to look at the pictures too, but it does tell you here that the cholera toxin causes the CFTR protein, which we're also talking about here, to stay open for too long, which causes chloride ions to leave, because that's the main thing it transports. Some sodium ions leave as well. And even though those are ions, those are also considered solutes. So remember, water always wants to go towards higher solute concentration. And so when those chloride ions and sodium ions leave the intestinal cell and go into the digestive tract, water is going to follow because it's going towards that hypertonic solution. Okay? So we're still working with a CFTR protein here. It's just going to do something different. So I want you to be thinking about what you said the CFTR protein did, or what effect the cholera toxin had on the CFTR protein, and how that CFTR protein is supposed to normally work, and how that is different in a person with cystic fibrosis, which is a genetic disorder. So normally, in people without cystic fibrosis, protein kinase A binds to the CFTR protein, opening it up. So CFTR, I believe, is cystic fibrosis transmembrane conductance regulator. It's a fancy name for this chloride ion channel. And that allows chloride ions to leave the lung cells. So now we're talking about the lungs. We're not talking about intestinal cells. And those chloride ions will enter your airways, which would be this area out here. Now, water's still going to follow, okay? Because chloride, even though it's an ion, is kind of like a solute. It's going to behave like a solute. So water wants to go to where there's more ions, where there's more solute. So water will leave the lung cells and go out into the airway space, okay? Like your trachea, or your bronchi, or your bronchioles. Water is going to follow those chloride ions out to go towards an area of greater solute concentration. And when it does that, that excess water is going to thin out the mucus there, and it's going to create this nice watery kind of mucus, which is what you want. That's what's normal. Does that make sense? Okay. So let's talk about genetic causes of CF. So in someone who has inherited 2 mutated copies of the CFTR gene, this disorder, cystic fibrosis, is an autosomal recessive disorder. Therefore, the people who show it are going to have inherited a mutated copy from both parents. What ends up happening, it's called a delta F508 mutation. Delta, remember, is like change in. F, as weird as it sounds, is the 1-letter abbreviation for the amino acid phenylalanine. Even though I know it doesn't have an F in the word, they sometimes use 1-letter abbreviations for these amino acids, and F is the 1-letter abbreviation for phenylalanine, which is shown here. So what happens in CF, is that this segment is deleted, which means there is no phenylalanine. Now, we already know from the first unit, you start messing and changing one amino acid from another, or in this case, deleting an amino acid from your chain, we know that in order for that protein to fold into the proper shape, it's got to have those amino acids and their R groups, or else certain bonds can't form. So when phenylalanine is missing, that is going to change the shape of that CFTR protein a little bit. And what it actually ends up mutating is the protein kinase A binding site. So remember, without that phenylalanine there, it's probably missing some kind of bond. So it's going to change the overall 3-dimensional shape that protein or polypeptide folds into. And the actual part of the CFTR protein that gets mutated is the place where protein kinase is supposed to bind. And if the protein kinase can't bind, then the chloride ion channel can't open. So notice how I want you to spell this out. If the chloride ion channel can't open, okay, then water can't leave the lung cells to go into the airways to thin out that mucus. And so the mucus is going to be really sticky, right? Really thick. And so that's what we should see on the next slide. Okay, so if you look back to your study guide for the potential essays, it says something about like contrasting, how the CFTR protein, I think, is normally supposed to work in CF versus how it works when it's mutated, and then how is that different from what it does in cholera. So if we backtrack to cholera, right, we know that the cholera toxin causes too much cyclic AMP to be made. What enzyme makes cyclic AMP? Adenylyl cyclase. And that would mean that adenylyl cyclase is staying active for too long. Because remember, that was one of our light switches we wanted to shut off. So try not to use the term charging. Okay, that was just my analogy with my sister's electric car. It's not really called that. It's whether it's activated or not. So for adenylyl cyclase to stay active, that must mean that an activated G protein was bound to it for too long. Some people in their case studies were saying, oh, the GTP causes adenylyl cyclase to stay active. Not directly, it doesn't. The adenylyl cyclase enzyme stays active because the G protein is active and stays bound to it too long. So then you've got to say, okay, well, why is the G protein staying active? Because it is unable to act as a GTPase enzyme to convert GTP to GDP, which would inactivate it. So if that step can't happen, then GTP stays bound to the G protein stays active and bound to adenylyl cyclase. Adenylyl cyclase therefore stays active, produces too much cyclic AMP. Cyclic AMP causes those chloride ion channels in the intestinal cells this time. So make sure you're talking about what part of the body we're in, to stay open too long. So too many chloride ions leave along with some sodium ions, and water is going to go to where there's an excess of chloride and sodium ions. Because sodium and chloride together make up sodium chloride, which is salt. So wherever salt goes, water follows. But the technical reason why it's doing that is to head towards the area with greater solute concentration. So in that case, you have too much water entering the digestive tract from the intestinal cells, which is what's going to give you that watery diarrhea. In this case, in the people with only mutated versions of the CFTR gene, that phenylalanine amino acid gets deleted. It changes the shape of the CFTR protein, deleting the protein kinase A binding site. Well, protein kinase A is what opens that chloride ion channel. So if protein kinase A can't bind to the CFTR protein, then the ion channel won't open. Chloride ions, I know this looks weird, but that's supposed to be the CFTR protein. Then the chloride ions won't be able to leave the lung cells and go into the respiratory tract, which means water can't follow, which is going to give them this very thick, sticky mucus, which they're going to have to take, enzymes in a nebulizer. They're going to usually put on this vest that kind of shakes their chest to loosen that mucus. And of course, because it's mucus, it is a breeding ground for bacterial infections. And this also can affect their digestive tract and reproductive tract as well. But for right now, we're just going to focus on its effect on the lungs. Nope. Not that. That's where we're going. All right, so this is the way I always end the unit. This whole unit is about cell-to-cell communication. And so we've already talked about how cells in your body, which you would be a multicellular organism, how and why your cells communicate with each other. So what we haven't focused on yet are the unicellular organisms, the organisms that are only made up of one cell, and how and why they communicate. Well, in all three of those, yeast, bacteria, and cellular slime molds, they don't have eyes. So they can't see how many others like them are around or where they are. So they're going to rely on chemical signaling to sense how many others are around and to convey, a distress signal if conditions are not good. So we won't get through all three of these today. We'll at least get through yeast. We may or may not get through bacteria, and we'll probably save cellular slime molds for tomorrow. Okay. So you don't have to memorize the name of that yeast, although you will see it in the animation I'll show you. It is the yeast Saccharomyces cerevisiae. If you've taken Spanish, I don't know if they teach that in high school, but cerveza is beer. So this is the yeast that is used to make bread and alcoholic beverages, because it's a fermentation process. And yeast are the ones that carry that out. All right, so for this, we've got to understand how yeast normally reproduce and when and if they would do this, be looking for a mate of a different mating type, why they would want to do that. So normally yeast reproduce asexually by budding, which means exactly what it sounds like. A little new yeast buds off of them and then separates and becomes its own little self-sufficient yeast cell. Because remember, these are only single-celled organisms. And so I think, I didn't see it, but a student told me that there was an episode of SpongeBob once where he says, "Are you capable of budding?" And the little SpongeBob buds off of his shoulder. That's budding, okay? A yeast cell buds off of a parent yeast cell, and then eventually this one will separate and become its own self-sufficient yeast cell. The problem with that is the offspring yeast cells are genetically identical to the parent, and the key to survival of a species is going to be genetic variety. So imagine I did the little SpongeBob rainbow, right? Genetic variety is the key to an organism's or a species survival. So yes, they may prefer to reproduce asexually by budding, but every now and then they're going to need to shake it up a little bit and introduce some genetic variety into their culture. Okay, so this is what I was just saying about genetic variety. We know that genetic variety is important for the survival of the species as a whole. So occasionally the yeast cells will want to find another yeast cell of an opposite mating type. Okay, now yeast cells do not identify as male or female. There are two mating types in yeast, and they are A and alpha. So we don't have boy yeast cells and girl yeast cells. So they want to find a yeast of an opposite mating type to combine with, but they don't have eyes. So how are they going to figure out where these other yeast cells are? By using chemical signals and receptors. Because pretty much in everything we've talked about in this unit, that has been our one constant that we kept hearing about over and over and over again. Chemical signals being released by cells and binding to a receptor, maybe on its own cell, maybe on another cell. Okay, so that's how they're going to figure out if there are any yeast cells of a different mating type nearby. Okay, so there are two mating types, A and alpha. Alpha to me kind of looks like a fish, depending on whether it's a skinny fish or a fat fish. So those are the two mating types. Oh, sorry, some of my line is covered up. Let me make that small, and then I'll make it big once we have that down. So when a yeast cell wants to mate, it has to figure out if there are other yeast cells nearby that are of a different mating type. So an A-type yeast will release A-type pheromones. Pheromones are chemical signals used to attract a mate. Okay, so there we have A cells, there we have alpha cells. The A cells are releasing these blue signaling molecules, and what we're hoping is that there'll be some alpha cells nearby, and alpha cells are going to have receptors for the A-type pheromones. So that's why you see some of these blue circles binding to the receptors on the alpha cell. Okay, so these pheromones will diffuse away from the A yeast and will hopefully bind to A receptors on alpha yeast if any are nearby. Okay, so we know that the cell that secreted those A pheromones, obviously they're going to be in highest concentration around that A-type yeast. But as with all things, we know that they can diffuse from an area of high concentration near those A yeast to an area of lower concentration. And so as they move further and further away from the A yeast, their concentration tends to go down a little bit. But what we're hoping is that at least some of those A pheromones will bind to receptors on the alpha type cell. And that's going to let the alpha cell know, oh, there's an A yeast nearby. Let me send out my alpha pheromones and make sure that they know that I'm here. Okay, so in response, the alpha yeast are also going to be sending out their alpha pheromones. And if they bind to receptors, also notice this. Notice where we don't have a side. We don't have a sign because it's a side because it's a circle. But when the A yeast release the A pheromones, notice how they're binding to receptors on the left-hand side of the alpha yeast. That tells the alpha yeast in what direction the A yeast is. Okay, same thing with the opposite. These alpha pheromones are going to bind to receptors on the A yeast cell on this side, which is going to let them know, okay, the alpha yeast must be that way. So alpha type yeast will release alpha pheromones, which will bind to receptors on A cells, just like I showed you in that picture. And so when you're practicing this process and writing this out, please don't shorten it and be like, well, this binds to this and vice versa. We don't write vice versa in any AP essay ever. Okay, spell it out if you want the points. Okay, so now if we think about those yeast, the A yeast had its pheromones bind to the left side of the alpha yeast, and the alpha yeast had its pheromones bind to the right side of the A yeast. So that kind of lets them know which direction these other yeasts can be found in. And so once those pheromones bind to their receptors on the other type of cell, then the yeast will start to extend their cytoplasm on the side of the cell, which I know is weird if it's a circle, but you know what I mean. On the side of the cell where the pheromones have bound to their receptors. So the A yeast, if you think about that picture from the previous slide, the A yeast are going to start extending their cytoplasm in this direction, and then the alpha yeast are going to try to extend their cytoplasm in this direction. Oh, and then I'll show you that again in that picture. So this cytoplasmic extension, like when the yeast cell goes from being rounded to having this cytoplasmic extension, they call that a shmoo. And the process of the cells growing toward each other is called shmooing. Now, people don't usually have trouble remembering that, but you can't get all the points in your essay for talking about shmooing, okay? Unfortunately, that's not the way this works. So you really do have to pay attention to the picture. You know what I'm going to tell you to do. Draw it out and talk through it. Explain it to your cat. Explain it to your dog. Explain it to your parents. They don't have to know what you're talking about. But if they can kind of follow you and not be that confused, you probably did a halfway decent job drawing and explaining it to them. I'm not saying you have to practice in front of your parents, but I'm just saying you want to be able to get some kind of feedback for how clear you're being in your explanation. So let's look at this diagram one more time. So the A yeast is going to release its blue pheromones, which will bind to receptors, in this case, on the left side of the alpha yeast. The alpha yeast is going to release its pheromones, which are going to bind to receptors on the right side of the A yeast. Now they know, hey, I know an alpha yeast is in that direction, and this one knows an A yeast is in that direction, and so this is when they start to extend their cytoplasms, which is called shmooing, and then their cytoplasms are going to fuse to form a cell that has both the A genes and the alpha genes. So this little creature is like it's a cartoon figure from I think the 50s or 60s called a shmoo. And so some nerdy scientist saw what they look like when this is happening and thought they looked like this cartoon character, which is where the shmoo term came from. Okay. Okay. So the result is the fusion or mating of two cells of opposite mating type. And that makes a diploid A alpha cell containing the genes of both cells. And the purpose of this process is genetic variety. So we're going to watch a little animation of that and then see where we're at. Just bundling stuff up for my people who aren't here. Okay. So see how they're growing towards each other? Okay. So I know that's kind of cheesy, but it's a good way to learn about schmooing, which is how yeast cells, um, when they want some genetic variety, can find yeast cells of another mating type to reproduce with. Mhm. All right, peeps. We are going to do a little bit on quorum sensing. Then we're going to do our lab in here. And then if there's time, we'll watch the quorum sensing TED Talk at the end. And that'd be great if we could do that if we have time, because then that would only leave slime molds for tomorrow. Okay, so this is our second type of communication. Or a second example of communication between unicellular organisms. And this is called quorum sensing. So first, in order to understand what this is, you've got to understand what a quorum is. So as it says there on the left, it says a quorum is the smallest numbers of members to be present at a meeting before its decision can be binding or valid. So oftentimes you hear about having a quorum, like maybe in the Senate or in the House or even at a city council meeting, that there has to be enough of the voting members present to establish a quorum in order to take a vote on something. So over here it says, well, let's get started now that we've got a quorum. So if there's 7 people on your committee, maybe you need a quorum of 4 people in order to take a vote on something. So the idea behind this is that bacteria are very small. They cannot do much harm to you unless they act as a group, which is a coordinated attack. But then again, remember that bacteria don't have eyes, they can't see. So if they are going to try to carry out a coordinated attack, they're going to need a way to figure out if there's enough bacteria surrounding them, especially of the same species, to launch a coordinated attack. Because one bacterium by itself is really not going to do much. Okay, so it says bacteria don't have eyes, so how will they know if there are enough bacteria around them to be able to launch a coordinated attack? The answer is quorum sensing. So by themselves, an individual, can't really do very much, and even if it releases these signaling molecules, they're not going to have any other cells around to bounce off of to let them know they're by themselves. And so if they don't get a response back, chances are they're all by themselves. But here, if they release these signaling molecules into the surroundings, not only will those signaling molecules bind to receptors on other bacteria, but they may even bounce off other bacteria and come back and bind to their own receptors. And so that's going to let bacteria know that there is a high population of bacteria surrounding them and that they do have enough to launch an attack. Okay, So that is where we're going to stop for now. We are going to watch a bacteria TED Talk, whether that's today or tomorrow, I don't know. I want to make sure we have enough time to do our case lab. So this just says forum sensing bacteria release chemical signals which can bind to receptors on other bacteria and even on themselves and let them know whether or not there are enough bacteria present to be able to launch a coordinated attack. Okay, so I'm going to give a copy of the lab to everybody. Yeah, just grab the pass. I'm going to give a copy of the lab to everybody because even if you're not the one tasting, I still want you to be able to get the data from somebody who is tasting so that you can see sort of how this miracle berry works. We had 11 people turn in permission slips and I have 10 tablets. So I wish that wasn't the case, but one person is unfortunately not going to get to do it, but I will pick that randomly in just a minute. All right, let's see what we got. Oh, I should have turned those lights on. So first we got to figure out which 10 of us are doing this, and then I think we're going to move our seats a little bit so that a person who's not tasting can sit with a person who is tasting. And this way it's not like a bunch of tables of just 2 tasters and everybody else just sitting around like, what do we do? So let me stop recording.